everyone. Sorry to not be there in person, but I was told the Zoom would be seamless, so I hope everyone can hear me. And let me know if you can't. I am delighted to introduce Caroline Escoubas, who is a postdoctoral fellow in my lab. So Caroline did her MD PhD training at Nice University in France in a very competitive MD PhD program. As part of that program, she was able to do her graduate work at Harvard School of Public Health, where she worked with Weill, studying mechanisms of longevity in C. elegans, a beautiful worm model system for studying aging. She's very passionate about psychiatry and has the goal to be a psychiatrist and researcher someday. And as part of that, when she came to the US, she joined my lab to study how the immune system impacts brain development and function. Caroline is a fearless scientist and investigator. She's an amazing multitasker as well. As evidence of that, the data she's presenting today was actually posted in a manuscript online just last night, and all while preparing this talk for you all. So I am very pleased to introduce Caroline and hope you enjoy what she has to tell you.
So thank you. Thank you, Anna. I'll shift to the presenter view, Anna. Okay, so thanks, Anna, if you can still hear us, for the kind introduction. And I'd like to thank the organizers and the committee for selecting my work today to present. And I'm very excited to talk to you today about our work really highlighting the complex interplay between the immune system and brain development in a mouse model. And so historically, the fields of neurology and neuroscience and immunology have been quite distinct, but I think there's a growing appreciation over the past couple of decades for interesting overlap between these two fields and interesting biology to discover. And as evidence of that, there are some really nice epidemiological studies that have been done that show that dysregulation of the immune system or certain immune-related genes may be correlated with certain mental health disorders or neurocognitive disorders such as autism, depression, and schizophrenia. And one very compelling clinical presentation of really this crosstalk between the immune system and brain function is this maternal immune activation model, where essentially pregnant mothers will develop severe bacterial or viral infection. And studies have shown that in these patients, their kids, the progeny of these mothers, are more at risk for developing certain neurocognitive disorders. And there's strong links for autism, for example, or schizophrenia. And basically what we think is happening in this system is that the maternal immune system becomes highly activated at a time where when the brain, the fetal brain is highly sensitive to environmental cues and to this immune insult. And that leads to an impact on the proper development of the neuronal circuits and later emergence of some mental health disorders. And so this is very interesting, compelling evidence from the clinical side. However, we still lack really good understanding of the mechanisms driving it and the cell types that drive some of these effects long-term on brain function. And one excellent candidate cell to study, and that we are particularly interested in studying in the lab, are the microglia. And so microglia are the primary immune cells in the brain, and they actually represent 10% of all cells in the brain. So it's quite a large number. They provide a lot of course, immune-related functions, but there is now a lot of evidence showing the importance of microglia in normal brain development as well. And so here I'm just showing you an example of a video recorded by a fellow postdoc in the lab, Haruna, and she did live imaging in the zebrafish brain, where you see a microglia tagged in green, which is highly motile, traveling through the brain and extending a projection, reaching out to engulf a neuron, which is tagged in red for engulfment. And so that's one of the functions that these cells can do in order to change the neuronal circuits. And so these microglia are highly sensitive, right, to the circuit and to their microenvironment. And in the lab, we're particularly interested in understanding what can change the microglia function, how they respond to changes in the brain, and how it later impacts brain function. And so to do that, we use a mouse model, and we study in particular the somatosensory cortex. And in the mice, the primary sensory organ are the whiskers, and they're so important to how the mouse relates to the world that there's a very defined, dedicated part of the cortex that these whiskers project onto. And in order to stress out the system and force the brain to remodel, we remove half of the whiskers on one side of the face of the mouse. And what it does is it forces the corresponding part of the cortex to remodel and readapt acutely. And what I'm showing you here on the left is the paradigm. So we perform this whisker deprivation on one side of the face at P2, so 2 days after the mouse is born. And then we study their brain 3 days later at P5. On your right side of the screen, you can see a synaptic staining. And in the top image, you see this nice barrel definition. So each little square actually corresponds to the projection of one whisker onto the cortex. And with whisker deprivation in the bottom panel, we lose this nice architecture. So we stress out the system. And using this paradigm, we then asked, how do microglia respond and adapt to this? And what we discovered is the emergence of this very special type of microglia, which I'm showing you an example of here. And so these microglia are almost absent in the control brain at baseline. However, they expand 20-fold after whisker deprivation once you stress out the circuits. And so for those of you who are not familiar, this is a confocal imaging of a single microglia, which is labeled in green. You can see my mouse. And in the background, in dark blue, you have the nuclei of the surrounding neurons. In light blue, over here, you have the microglia own nucleus. And what was really striking about these microglia that emerged after stress is that instead of looking on the left, sorry, the mouse is not great, very ramified with all these little projection, which is kind of their baseline state at this age, this microglia was actively eating other cells in the brain. And so what you can see here is on the top, the microglia is extending a projection and wrapping around another cell. And so in dark blue inside, you have the nuclei of a cell that is being engulfed and digested by the microglia. And what was really striking, in addition to just seeing them engulf, is they tend to engulf and eat multiple cells at the same time. And you can see at the bottom, it's extending another projection to wrap around the second cell. And we did further work showing that these are actually neurons that these microglia are eating. And when we look a little bit more closely at the gene expression profile of these microglia using single-cell RNA sequencing, we saw that they overexpress all these specific genes that are in the type 1 interferon pathway. And that was highly surprising to us because the type 1 interferon pathway is actually an canonical antiviral pathway. So you would expect it to be activated during a viral infection. However, you would not expect cells to activate this pathway under sterile baseline condition. So this was really puzzling, and we wanted to understand why these cells, these microglia that are eating neurons in the cortex, activating this pathway specifically. And so to do that, we decided to block the pathway. And so type 1 interferons, which are comprised of interferon alphas and interferon beta, signal through their receptor, which is composed of two subunits, IFNAR1 and IFNAR2 proteins. And luckily, we have mice models available which are mutated for the IFNAR1 protein. We have either a global knockout, meaning that all the cells in the mouse lack this protein, or we have a microglia-specific knockout where we delete this protein only in the microglia to try to understand its function. And so we looked at these mice and we looked at their microglia in the cortex. And what we saw was a very striking phenotype where these microglia looked very dysmorphic. They looked very weird all of a sudden. And so on the left here, you have on your right, you have confocal images of microglia from a wild-type animal, which is actively engulfing another cell. So this is typically what you would expect. You have the microglia in green, the microglia on nucleus in light blue, and it has a phagosome, so it's wrapped around another cell and is in the process of digesting it. On the right side, you have a microglia when in an IFNAR1 mutant. So now when we block the type 1 interferon pathway. And now we see this microglia with these enlarged compartments, much, much bigger than the microglia on soma and on nucleus size, with very thin walls. Those are actually phagolysosome. We can see that with our CD68 staining in red. And we also see DAPI signal, meaning that there's an engulfed cell in there. So what we think is actually happening that leads to this phenotype is that these cells, they reach out, they engulf a neuron, but they're unable to properly digest it and properly resolve this phagocytosis event. And they get stuck with these huge compartments and cannot eat the neurons they're supposed to eat. And so that's the model we started to develop, where we think under stress in the circuit, certain stressed out neurons are being targeted by these very specific microglia that emerge in these conditions and that activate type 1 interferon pathway specifically, and that eat these neurons to remove them from the circuit. So then we wanted to ask, how important is it overall for brain development and brain function? And here, I just want to pause for a second and really highlight that studying the studying the somatosensory cortex is particularly important. Studying how it properly develops, we think is really important. As clinicians, you're probably very familiar that one of the hallmarks of autism is hypo or hypersensitivity to outside stimuli. And there's actually now very interesting studies in the mouse showing that some of the genes identified in autism patients are genes that are actually important in sensory neurons and proper connectivity in the somatosensory circuit. So what can we learn in our model from this? And so what we did first is look a little bit later, 10 days later. So the mice are now 2 weeks old. And we looked specifically in the somatosensory cortex, and we stained for a number of excitatory neurons and inhibitory neurons marker, and we counted. And here I'm showing you images from a wild-type animal and an IFNAR knockout animal, which is the mouse that we've identified that microglia are unable to properly eat the neurons that they should. And what we saw is an increase, and here I'm showing you specifically of one marker, CTIP2 positive excitatory neurons. So now we see changes in the relative abundance of certain subtype of neurons. And overall, with these different markers we looked at, we saw that there is a slight increase in the number of excitatory neurons and a slight decrease in the number of inhibitory neurons. So there's a shift in the balance between excitation and inhibition in these mice. And the last question we wanted to ask is, does this have any relevance at the behavioral level? Is it important for the proper function of the somatosensory cortex? And so to do that, we perform what we call the whisker stimulation assay, which is a very simple assay where we stimulate the whiskers on one side of the mouse repeatedly, record the mouse, and then assay how the mice respond to this stimulation. And so, let's see the video. Okay, so here on your left, you have a wild type animal. So I'm stimulating the whiskers and it's pretty much unbothered by the stimulation. However, when we perform the exact same assay in now our mice that are mutated for the type 1 interferon pathway, and this is true for mice that lack the type 1 interferon pathway in the whole, all the cells or in the microglia only specifically, we see that these mice respond very differently to the stimulation. If I can show you again. They're very avoidant. They run around the cage. They try to shield their whiskers away from the Q-tip, and they even become aggressive towards the Q-tip. So they're highly reactive. And we think this really reflects this tactile hypersensitivity phenotype, which we think results from the loss of type 1 interferon signaling in the microglia specifically. And we quantified this with a composite score, which scores the degree of each of these behavior, the fearfulness, the avoidance, the aggressiveness, and then confirm that indeed these microglia have a stronger reaction to this stimulation. Sorry, it's not moving. Okay. So now we think this is what we discovered during development in this using these mice models, is that during development, we think this is something that happens physiologically with very low levels of interferon in the brain. When there's a stressed out circuit and a stressed out neuron, it releases some signals that we are currently working on identifying to tell the microglia to eliminate them from the circuit. And so we think this microglia activate the type 1 interferon pathway in order to target these specific neurons for elimination in order to allow for proper brain development and proper brain function. In our results, we show that when we block this pathway and when we block the ability of the microglia to mount an interferon response, they're unable to properly digest neurons from the circuit. We see an accumulation of stressed out neurons, changes in the relative abundance of excitatory and inhibitory neurons, which later has an impact at the behavioral level. And we think this is giving us maybe insight into other disorders. So we're specifically studying this type 1 interferon responsive microglia during development. However, there's a number of transcriptomic studies out there showing that this specific population is actually present in a number of other disease models. One of the main obvious ones is any viral encephalitis models. And for example, COVID encephalitis models have been shown to mount microglia, mount a type 1, very strong type 1 interferon response pathway. We've actually shown it in the lab on some slices. We see this microglia. But we also know from other people's work that it's present in Alzheimer's disease models and in aging. So, and the big gap in the field was we did not really understand what these microglia were actually doing to the circuits. So we're interested in asking if what they're doing during developments is true in other models, and could that impact the disease progression of some of these other models where these microglia emerge. And with that, I'd like to thank all the people that contributed to this work, the whole Malofsky lab, and particularly Anna for her mentorship. And just want to highlight the key contribution of the three people at the top. Leah Dorman, who was a graduate student in the lab and who started this project, did all the bioinformatics and is still working with me. Fee, who was a graduate student too, and who now moved on to a postdoc. And Christian, who's a talented lab technician who's been working with me for the past 2 years. And with that, I'm happy to take any questions.
Do we have any questions? First is, what do you mean by a stressed neuron?
That's a great question. So I didn't want to detail here. The one marker that has consistently tracked over all our models is these cortical neurons with double-strand DNA breaks inside of them. And now we're trying to do more work to really understand what exactly these neurons are. We think there's a couple hypotheses. One hypothesis would be a hyperexcitability issue. There are papers out there linking double-strand DNA breaks to hyperexcitability in neurons. And so we're investigating those things.
Thank you. Very nice talk. I have another question about specificity. As I understand it, there are models of immune inflammatory related mechanisms in, as you pointed out, autism, depression, schizophrenia. There are microglia all over the brain. Why does one person get autism, one get schizophrenia, one get depression?
That's a great question. Maybe because of it, you need multiple insults. Maybe you need, maybe this plays a small part. A patient is more sensitive to mount a type of interferon response in microglia, and that plays a role. I'm sure there's lots of, you know better than me, but genetic factors, environmental factors that also play into the development of these disorders.
Thanks.
Thank you for that talk. It was fascinating and way out of my area, but I understood it, which is fantastic. And I have a question about the hypersensitivity, the behavioral data you were showing where you poked the whiskers with the Q-tip. Would the same thing be true if you had poked like the hind leg? Or are they hypersensitive to everything or just to the whisker?
That's a great question. My guess would be yes, they probably display other phenotypes of hyper or hyposensitivity. We didn't know when we started looking, and we're interested in maybe trying a couple different assays to maybe see if we can, other, at least in the tactile, like other tactile stimulations, see if it remains true. Yeah, we have a couple different assays in mind, actually.
Okay, thank you.
Do we have? Oh, sorry.
I also not in this area at all. I'm pure human person. A very quick question about the hypersensitivity as well. I'm assuming you spend a lot of time observing the mice. So is that difference from the baseline? So in the baseline, they don't react like that. They don't become aggressive. So how do we know?
In a wild type animal?
In the IFNAR1 mutant.
Oh, their baseline level of anxiety and stress. So we didn't do those assays in the lab. I haven't noticed anything different at baseline. So before we do the assays, we habituate the mice to the cage and to the environment to try to decrease the amount of stress from the assay. And usually the mice just kind of sit in a corner, explore a little bit, and then, I personally haven't seen much at baseline in this assay at this age. That's not to say that there might be other anxiety-related phenotypes maybe later on in adulthood.
Thank you so much. Great talk. Thank you.
I'm seeing a question from the chat, which I can read here from Maga. She's asking if they follow the progeny, if we follow the progeny of these hyperactive mice to see if any of these phenotypes might be heritable.
Yeah, no, that's a great question. We haven't done any of those experiments. Yeah, I don't know if we would necessarily see a phenotype in these mice at baseline, but maybe if we stressed out the system, stressed out the mother, then we might be able to see a phenotype, because we think they wouldn't be able to mount, an interferon response appropriately. Maybe we see a phenotype in the progeny.
We could imagine that a viral response in the mother might directly impact a fetus and cause similar kinds of microglia to emerge. But whether the phenotype passes from mother to child, our model is that this is kind of a hit and run, right? So a viral infection might acutely cause these microglia to show up and get rid of neurons.
Yes.
Wonderful talk. Congratulations on the award. Really exciting to identify sort of a new and surprising aspect of biology. And I think beautiful talk, both visually, by the way, I love that, and the work. I want to push a little bit on the issue around kind of the specificity of these kinds of early insults. I think it's a really, key question right now, as to whether, kind of any insult in early development can lead to any neurodevelopmental, any psychiatric disorder, or whether or not we see some specificity. So my read on the literature is that the link between immune mechanisms and autism in utero is overall pretty weak. That the epidemiological data does not support viral infection in the way that it does in schizophrenia. When you look at the genes and you annotate them, there are 255 of them, we don't see a strong signal, at least for what we know, and there are limitations, obviously, to those annotated approaches. We look at transcriptional data. There's a little bit of a suggestion around glia, but it wasn't kind of a strong signal in terms of early development. And then you do see some in postmortem brains, but there's not a corresponding signal in common variant genetics. So I guess the first question I have is, do you read the literature differently? And the one other one is the maternal immune activation mouse model, which is based entirely on face valid derangements in social behavior, which unfortunately is kind of a tenuous way to draw a link to a human neuropsychiatric disorder. So do you read the literature differently? Do you think, am I missing stronger evidence for this mechanism being directly relevant to autism? Or is it that, these are very early days, there's a range of interesting things? And if that's the case, is the evidence stronger around, for instance, schizophrenia or AD for the role of these molecules and potentially this mechanism as a pathogenic feature?
No, I think you make fair points. I personally read the literatures as like you say, it's the early days. It's giving us a hint that some of these factors might play together. I think one key thing that we might be missing, and which really we think is highlighted in our work, is we think some of these responses are very transient, and we might miss a lot of what is actually happening. In our model, I didn't specify it, but these microglia population turn on and off within 24 hours, we think. So we don't see them at baseline. They're really hard to detect when you just sequence at baseline, even different models, and you really need to catch them at the right time point with the right insult, and which is how we found them here. So I think there's still a lot to understand about mechanistically, but maybe once we know a little bit more mechanistically what these cells are actually doing, then maybe we can ask more specific questions, like to some of these models.
I like that answer a lot. Thank you very much.